ladies and gentlemen. My name is Simon Gaskell. I'm President and Principal here at Queen Mary University of London, and I have the pleasure to welcome you all to this evening's inaugural lecture to be given by Martin Knight, Professor of Mechanobiology in our School of Engineering and Materials Science. And we're privileged this evening to have his distinguished external chair for the event, Professor Dan Bader. Now, I say external, but really he's one of us in two senses. A former member of staff, a former academic colleague here at Queen Mary, and still, in fact, a visiting professor. Dan Bader studied physics at Liverpool University, followed by an MSc in medical physics and a PhD at Southampton University. He moved to the Oxford Orthopaedic Engineering Centre at the University of Oxford, where his research focused on engineering aspects of pressure ulcer prevention. He later moved here to Queen Mary as a lecturer in biomaterials and was one of the core research staff in the Interdisciplinary Research Centre in Biomedical Materials. In 1999, he was appointed Professor of Medical Engineering in the then Department of Engineering here at Queen Mary, a post that he held until 2011, when he was appointed Professor of Bioengineering and Tissue Health in the Faculty of Health Sciences at the University of Southampton. Professor Bader is widely known for his contributions to the translation of bioengineering solutions in the prevention of pressure ulcers, biomechanical conditioning of cell-seeded constructs for tissue engineering, and cell tissue biomechanics in health and disease. Professor Bader, thank you very much for coming this evening to chair the event. May I now hand over to you.
Well, it's a great pleasure and honour to be invited to share with you this joyous and celebratory event for Martin. It was almost, I think, 24 years ago that Martin appeared in my office in the IRC building, which I gather it's being reconstructed, and came in and we discussed the possibility of him joining us as one of the 10 PhD cohorts. I think it was our second cohort in the IRC. And we discussed what he was interested in, and he explained, I'm really interested in cartilage and joint degeneration. So I said, well, that's convenient because both David and Lee and I were interested in that ourselves. I said to him, well, why are you interested in cartilage? And he said, well, I've had a family member who's been involved in cartilage for many years, and I looked down at his CV and saw that his home address was Winchester, and I said, you're not related to David Knight, are you? And he said, yes, that's my dad. And David was a supervisor of mine when I was doing my PhD in Southampton. So clearly nepotism at the time was live and kicking. And we appointed Martin, and I have to say it was a very, very good decision in doing that. He embarked on a project which was new to us in looking at cell mechanics using some techniques such as imaging techniques, confocal microscopy, that we hadn't employed before. And we were also involved in tissue engineering, which was a new subject and was given a big hype in terms of being the way to repair cartilage damage, early cartilage damage. I realised at that stage, Martin embarked on it with a load of enthusiasm and a large amount of skill as well, and technical prowess. But during that time, he gained a reputation for having hollow legs. And I realised he used to run around the badminton court and the football pitch with not much skill, it has to be said. And I realized I wanted to know why he had hollow legs. And we went to a meeting in the United States where Sarah was there, his future wife. And in typical American fashion, we went down to breakfast and there was a mound of pancakes that were appearing on our plates with maple syrup and fruit. And I thought I was a big eater, but by the time I'd finished my plate, Martin had finished his own plate, plus most of Sarah's as well. But anyway, as we moved on, Martin became a very independent researcher and his expertise was well recognised internationally. And he had lots of links all over the world, but particularly in New Zealand with Professor Tony Poole, who at the time was the pioneer in identifying the cartilage cell, the chondrocyte, plus the matrix around it, the so-called chondron. And I know Martin's worked with that New Zealand group for many years. I'm pleased to say that also an ex-PhD student, Sue McGlashan, is now a reader in that department and still maintains links with Martin. So without further ado, I just want to say what a loyal person and also a very nice person, as well as being a very talented person, Martin is. So if I can now pass the baton on to my co-supervisor, David Lee.
Thank you very much. I mean, it really is a delight to be able to be involved in this event, and I'm very pleased that Queen Mary has relaunched its programme of inaugural lectures, which had fallen fallow for a little while. And I do think the notion of inaugural lectures are incredibly important. It really is a significant and important accolade to be able to attain the position of chair. And I think it's something that we should be acknowledging, recognising and celebrating. And there's no better way of doing it than through an inaugural lecture. So actually, this is the first time as head of school that I've had the pleasure of chairing an inaugural lecture. But it's a double pleasure because Martin was my first PhD student, along with Dan. So there's a sort of double rite of passage here, in that there are several stages during an academic career where you really feel that you have attained a new step. One of them is, of course, becoming a chair, but another one is when your first PhD student themselves gains a chair. So it's doubly fantastic occasion for me. Now, one of the things I would like to say about Martin is that in many respects he has adopted a very old school approach to his academic activities. And I mean that in the very best way. He's always been somebody who's very careful at the attention to detail, making sure that he does something extremely thoroughly. And that's a very, very important attribute. And indeed, myself and Dan were talking about, in the early days when Martin was doing research, we had a colleague, a collaborator that we worked with quite closely at Duke University. I shall not name him. Those of you who know the area will know him rather well. And of course, when you're in the same research area, there are ideas, there are questions that arise that a number of groups follow. And it used to be a standing joke that this colleague in Duke would get a bit of work done, get a paper out, and then Martin would do the work properly. And he would pay attention, he would deal with the detail, and the work that he would produce would be another order of quality. So that old school approach, I think, is very, very laudable. But what Martin has been able to do is combine it with a thoroughly modern approach of a modern academic, with all of the things that are thrown at us in our academic lives today, taken them on, delivered, for example, exceptional areas of education to students at all different levels, tireless in pursuit of public engagement activities. And to combine that with the attention of detail is rare. So the other thing which people may not know about Martin, and again is a very thoroughly modern thing, I receive, and maybe some of you who are in the academic field receive these things academics say, little cartoons. And I saw one the other day which said, how do academics deal with the work-life balance? You just make your life your work. So that's all that you do. And actually that's not right. we should, as academics, be having a good work-life balance. And Martin has done that. Many of you may not know, but Martin, for many years, has worked as a 0.8 FTE academic, effectively working 4 days a week so that he can commit to family life and being very fully engaged with his family. And that is a really brilliant thing to do. And as his head of school, I've never seen any diminution in the amount of work that he does. Indeed, he seems to manage to do a lot more in 4 days a week than perhaps some other colleagues do in 5. So I think that's a really important, it shows Martin's commitment to his work and also to his family life. And I'm sure that today Martin is very proud of his family who are all here. And today, of all days, I'm sure that the family will be deeply proud of Martin as we hear his inaugural lecture. So I've said quite enough. I think it's time now to give the floor to Professor Martin Knight.
Thank you, David, and thank you, Dan. Thank you for getting me here as well. We should probably stop now because that was such a nice introduction, but we'll keep going. It's brilliant to see so many people here, familiar faces, friends and family and colleagues, students, even some ex-students I think that we have here as well, and also people I haven't met yet. So you're all very welcome. I'm going to begin this evening with a clip from a short film that was made about me for the YES programme. And the YES programme is an educational resource that goes out to schools across the country, linking the national curriculum with real jobs. And this particular programme has been seen already by 65,000 young people since the programme was released in 2013.
Hello, my name's Dr. Martin Knight. I'm a medical engineer here in a university. This is Queen Mary University of London. Medical engineers use maths and science to try and solve medical problems. And the particular problem that I work on is what happens if you have an injury or a disease to your joints, like your hip or your knee.
Okay, so you can see from that that I'm a medical engineer, also known as a bioengineer or a biomedical engineer. And bioengineers are perhaps best known for developing medical implants, biomaterials, prosthetics, and diagnostic technology. But bioengineers also use engineering approaches to understand how our bodies work in both health and disease. And it's into this area of bioengineering that my own research best fits. So as you've heard, I was made Professor of Mechanobiology, and mechanobiology is the study of how biology responds to mechanical forces. And that might be a physical response, so looking at the strength of bone or the stiffness of ligaments or the stretchiness of our skin, and we call that biomechanics, or it might be a biological response. And I think it's important to realise that our bodies are constantly exposed to an ever-changing mechanical environment. So as I'm standing here in front of you, my blood is pumping and my lungs are expanding and inflating and my muscles contracting, joints moving, and my skin is stretching, my skeleton's being loaded, my stomach's probably churning. And how our bodies respond to those mechanical forces is really important. So I've put up here 4 images. This is a bit of a copy from the news quizzes, or have I got news for you? And the question is, what links all these 4 images? And the answer is that they're all examples of where mechanical loading causes remodeling of bone. So the astronaut there, when astronauts first went into space, they suffered from osteoporosis. Their bones started to break down and waste away because they weren't able to load their bones due to the low gravity. The same also occurs for paraplegics. So if you're in a wheelchair and you can't load your legs, then the bone begins to waste away. The image on the bottom there is an X-ray of a total hip replacement. And because the metal that is used in a total hip replacement is so much stiffer than the surrounding bone, this can shield the bone from the normal forces which keep that bone healthy. And consequently, the bone around that implant can begin to waste away. The odd one out is perhaps Andy Murray at the top there, because if you were to X-ray his forearm, you'd see that the bone in the hand that he holds the racket with has a higher density, is much thicker bone than in the other hand. So there the bone is remodeling and building up due to the higher forces, whereas in the other three, the bone is beginning to break down due to the lower forces. And this has been known about for some time. So this person here, Professor Wolf, first identified this back in 1892, that bone responds to the mechanical forces to which it's exposed. And since then, we know that the same is true for many other tissues in the body. Our tissues respond to mechanical force. But what we don't really understand is how those tissues can sense that mechanical force, how the cells that make up that tissue can sense it, and that is really important for keeping that tissue healthy. So we can look at mechanobiology across a range of length scales, and this is actually a slide that I have copied and adapted from Professor Bader's many years ago. It used to be a little sort of slide that was made of plastic and glass, but I've adapted it. So we can do this mechanobiology that ranges from the whole body right through to the protein level at a nanometer scale. So we have this hierarchical approach, and probably most of my work fits in the middle, looking at cells and tissues and how they respond to mechanical forces. So throughout my work in mechanobiology, I've looked at mechanobiology in the context of disease, and most of the work has been looking at arthritis and osteoarthritis. But more recently, I have worked with cancer, tendon injury, also a bit on nerve injury, and very recently looking at mechanobiology and aging. And this has all been made possible by the fantastic collaborative networks that we have here at Queen Mary, linking colleagues both in science and engineering, but also through to the medical school. And what I wanted to say at this point is that for me, understanding mechanobiology in these disease conditions is an interesting and stimulating puzzle. It's a real sort of interesting thing that I work on. But we must never forget that the people that suffer from these conditions and their families and friends, these are serious, debilitating conditions. And it's important that we remember that. And of course, that is our motivation. So as you've heard, I joined the IRC in biomedical materials back in 1993 as a PhD student supervised by Dan and David here. And at the time, the IRC was headed by Professor Bill Bonfield and was a fantastic centre training PhD students as well as producing very high quality, high impact research. And after my PhD, I got a personal fellowship to work, continuing some of the work from my PhD, and I continued that for 6 years. So during the PhD and subsequent work, I developed a love of confocal microscopy. And at the time, confocal microscopy was a very new technique, and still trying to learn a lot about it. And the point with confocal microscopy is that it allows you to take three-dimensional images of living cells. So the image on the far side there is a single cell. It's about a hundredth of a millimeter in diameter, and we can see and rotate that to look at the cell and see how it behaves. The other image shows a time-lapse movie. So this is a 4D movie showing a 3D image of the cell as it moves across a biomaterial surface. So you can see it moving and responding to that biomaterial surface. So I also, as well as a love of confocal microscopy, met Sarah, who's looking very embarrassed here, and fell in love with her. And this is Sarah on Professor Bader's computer. And she was doing her PhD, also supervised by Professor Bader, looking at the effect of mechanical force, at looking at pressure on skin and soft tissue breakdown in the context of pressure ulcers. and she needed healthy volunteers, and of course I was super keen. So this is a picture of me. It actually appeared in one of her papers in the Journal of Applied Physiology. This is me with her rather torture contraption putting force onto my bottom. And then what Sarah had to do was to mop the sweat from my bottom and analyse that to look at the breakdown of the tissue. So after that, there was nothing for it, and we had to get married. I should say Dan also volunteered, but I think he was already taken. So there's lots that I'd like to be able to tell you tonight, but I've had to put together a bit of a story. But I just want to illustrate a few of the things that I haven't really got time to tell you in detail. So I work with a company, and I'll be honest here, it's the company is involved with my dad, who helped set this up, and I've been involved with this, looking at the biomechanics of a novel silk implant. So this is Orthox Limited, and they've just got this material, so it's going into into clinical trials with patients at the moment. So we were involved in testing the biomechanics of this implant and looking at how it compared to the biomechanics of the native meniscus. I've also worked with various urology companies looking at implants that go into the urological system. So these are stents that go in the urethra and become blocked with these salts and incrustation. And we worked with the companies trying to improve the design of these implants and also look at novel ways of removing the incrustation without having to operate on the patient. Throughout my career, I've done a lot of work on cell mechanics. And the way you do cell mechanics, or one of the ways, is to get a very skilled student who can make these tiny glass pipettes and can attach those to an individual cell, so you see that in the movie here, and then suck the cell into the pipette, and then with the deformation of that cell gives us an idea of its mechanical properties. And lastly, I was also involved in some work looking at the biophysics of the urine stream. And so this was the first time that anybody had been able to understand the mechanics of why your urine appears to come out in that strange spiral, and could we use it as a diagnostic parameter. My children, or it's disappeared, might recognise the sophisticated measuring instrument that was down the side of that image. But most of my work has been on articular cartilage. And articular cartilage is a real engineering tissue. So I'm an engineer by background, and cartilage serves two very important engineering functions. It provides a low-friction bearing surface, which allows your joints to move smoothly. And it also protects the underlying bone from high stresses. So this is a cross-section through cartilage. It's a microscopy image, and you can see the cartilage is about 0.5 millimeter thick. The surface of the cartilage is at the top of the slide there, and then at the bottom is the underlying bone. And in the cartilage are the cells, which we call chondrocytes. And they're responsible for producing all the rest of the cartilage. The 70% of the tissue is not made of cells. It's made of something called the extracellular matrix. And that's composed of various proteins, collagen, something called proteoglycan, and water. And it's this extracellular matrix that gives the cartilage its mechanical properties, allows it to perform in the demanding environment of the joint. And the reason for looking at cartilage is really because of this disease, osteoarthritis. It affects around 8 million people in the UK and results in stiff and very painful joints. And currently there is no cure for osteoarthritis. And so what happens is the cartilage breaks down, and then frequently the option is to give them a fantastic implant. It's a bioengineering implant, as you see in the schematic there for the knee joint. But there are problems with these implants. And so I'm motivated to see if we can understand osteoarthritis and then potentially develop new treatments for this painful and debilitating condition. And to understand osteoarthritis, we've first got to understand about cartilage. And the important thing with cartilage is to realise that it's a constant state of balance between synthesis of new tissue and breakdown of the cartilage. So this is balanced, and this is made possible by mechanical loading. So the cartilage responds to mechanical loading. Just as I showed you with bone, so does cartilage respond to loading. And if the loading increases, then the balance switches a little bit more towards synthesis of new cartilage. And if, conversely, the loading decreases or disappears, then the balance switches a little bit more to breakdown. And if it disappears completely, then the cartilage really becomes very soft and prone to breakdown. So what we knew is that cartilage responded to mechanical loading, but we didn't know what the mechanism for this was. And so this has been the focus of much of my research. And more recently, I've been interested in the role of a tiny cellular structure in this process called the primary cilium, or cilia in plural. And those of you who understand any Latin, Uncle Roger, wherever he is, will realise that cilia means eyelashes in Latin. And I think that's right. And we'll see that the structure of primary cilia actually resembles a single eyelash. So it's a very long, slender structure. So I've got here a picture of motile cilia, and motile cilia probably are the cilia that most people are familiar with. So these are the cilia that move fluid around in the lung, help to clean the lung, and also responsible for moving fluid in other applications. And what I'm going to do now, I don't think has ever been attempted at an inaugural lecture, and probably never will again. We're going to get you, I'm going to get you, hopefully, to be motile cilia. And so what I want you to do in a minute is to raise your arms, and we're going to do a sort of Mexican motile cilia wave. And if I was to introduce right at the back row there, bucketfuls of mucus, something that I have occasionally been tempted to do in an engineering lecture, then you as motile cilia would be able to beat and move that mucus down to the front. And the other reason for doing it, sorry, I should say up there is a movie. You can see the beating cilia, which was provided by Dr. Amelia Schumacher. But the other reason for doing it is for Professor Beda here, who's probably looking a little bit worried. So Professor Beda has always been a Chelsea fan. And throughout my PhD, I had to monitor the success of Chelsea very carefully. I even would have my mother ringing me up to tell me if Chelsea had lost, so that I knew to avoid Dan in the morning. Anyway, Chelsea are due to play, I think, a football game somewhere in London tonight, and normally Dan would be there on the terraces. So I managed to get some very rare footage of Dan and his chums at Chelsea doing the motile cilia Mexican wave. So to make Dan feel a bit more at home there, we're going to do this motile cilia Mexican wave. So can you all stick your arms up in the air? Thank you very much. And you've got to do a sort of Mexican wave starting from the back, but if we're going to make this physiologically relevant, then it's got to be a sort of swirling Mexican wave. Come on, down from the back. Imagine that mucus is coming. Brilliant. Oh, and I'll just start this, so we're in time with Chelsea. There they go. Look at that. And there's Dan, if you can just see him. Thank you. I think we can say you are cilia. Sillier than I expected, anyway. So that's motile cilia. The actual cilium that I look at is called a primary cilium, or primary cilia. And these differ from motile cilia in that there's only one per cell. And also they lack the molecular motors which causes a motile cilium to move. So these are a single cilium, primary cilia, and they're about 1 to 5 microns in length. So the whole cell is about 10 microns in diameter, so 1/100th of a millimetre, and primary cilia are about 1,000 to a few thousandths of a millimetre in length. And they're very long, slender structures. If you cut them as a cross-section, you see they have rods of a protein called tubulin that go along the length of the cilium. And cilia are actually our oldest known cellular organelle. So motile cilia were first described back in 1675 by this person here, whose name I won't try and pronounce. But he described them as incredibly thin feet or little legs which were moved very nimbly. And I think that's a nice explanation. I'd like to be able to put something like that in one of my papers. But primary cilia were actually discovered sometime later, in 1898, and the term primary cilia was only coined in around 1968. So this is a classic picture of primary cilia. This is in the kidney, and here the primary cilia stick out into the lumen of the kidney, and they bend in response to the fluid flow. So they are mechanosensors, sensing the movement of urine in the kidney, and that's very important for keeping the kidney healthy. But primary cilia are not just found in the kidney, they are found throughout the body in all sorts of different tissues, in the skin, the bones, the nerves, blood vessels, stomach, in the cartilage, which is what I work on predominantly, and also in tendon, and as we'll see, also in stem cells. And yet, there are many people who don't really appreciate that they exist, or were at least sceptical. I can remember when I first told my colleagues, John Priestley and adenomyocarditis, about primary cilia in nerves, they laughed, but in a sort of funny kind of way, and I'm not quite sure what they were getting at. But primary cilia are there. And I was sent this picture of a primary cilia. So some people think that they're rather like the fictitious Loch Ness monster. They don't exist. But I can assure you, and certainly if you've looked down a microscope and seen primary cilia, they definitely exist. So the reason that we have recently begun to know so much about cilia stems from the discovery of these set of diseases called genetic ciliopathies. And these are defects, these are diseases associated with a defect in the genes that code for some of the proteins that build primary cilia, or that are associated with primary cilia. And the most widespread of these, and well known, is polycystic kidney disease. So this is a disease that is dependent on the primary cilium. And so I wanted to look at primary cilia and to find out if they had any function in articular cartilage, and particularly whether they were involved in sensing that mechanical environment to which cartilage is exposed. And to do that, I teamed up with Tony Poole in New Zealand. I'd already been out to visit Tony previously, as Dan had said, to work on something different in cartilage. But Tony was actually one of the first people to describe primary cilia in cartilage and other connective tissues. And this is, the bottom image is an electron microscopy image showing a primary cilia from one of Tony's papers. And I put this graph up here. This shows the number of journal papers, publications, that have been written about primary cilia versus the year. And the arrow indicates Tony Poole's paper back in 1985. And you can see there was very little work going on on primary cilia back then, and people thought Tony Poole was mad. It was the Loch Ness monster syndrome. They didn't believe primary cilia existed or that they had any function. And it was only when those genetic ciliopathies were identified that the interest in primary cilia really took off. So I travelled to New Zealand again in 2006 on a Royal Society grant to work with Tony Poole. And this was made possible partly by the generous funding of the Royal Society, but also by the arrival of Thomas, who's somewhere back here. There he is. And Thomas is my youngest son, and this meant that Sarah was on maternity leave, and so we could go as a family, and I worked in the lab, and Sarah did home education for the children, and then we travelled a bit around New Zealand. And I was working with Sue McGlashan, who Dan mentioned. So Sue was a PhD student here, and then I helped her to get that position in Tony Poole's lab in New Zealand, and she's working on cilia as well. And the other person there is Cynthia Jensen, and Cynthia actually published the third ever paper to mention primary cilia that's in the literature. So she's one of the original greats in primary cilia research. So I worked with Tony and Cynthia and Sue to try and understand more about primary cilia. And here they are. So this is primary cilia in cartilage cells, and this is a confocal microscopy image. So you can just see these tiny little red sticks. So I've stained it with a dye, and that's why they appear red. Blue is the nuclei of the cell, so each nucleus is shown in blue, and you can see that every single cell there has a primary cilium. So this is a confocal microscopy image. And this is our latest microscopy tool. This is a super resolution microscopy image. So super resolution microscopy won the Nobel Prize for Chemistry back in 2014, and we now have our own super resolution microscope here in the university. And this allows us to really look at unprecedented detail at primary cilia and other structures within cells. So we wanted to look and see whether primary cilia were involved in sensing the mechanical environment to which cartilage is exposed. And to do this, we took some cells that had primary cilia and also some mutant cells where the cells were unable to express primary cilia. And what we did then was to mechanically stimulate those cells. And at Queen Mary, we have fantastic facilities. We've just had a new investment of funding, which gives us even better facilities. And so we have one of the best reputations for being able to do all sorts of mechanical tortures to cells. So we can squash them and stretch them and shear them and poke them and pull them. And this is an image showing cartilage cells that are being squashed backwards and forwards in a tissue here. So we did that to our cells that had primary cilia and also to the cells that didn't have primary cilia. And what we were trying to understand is the biological signaling pathway through which a cell senses that mechanical environment. And this is what if you look up a signaling pathway, these are the sort of pictures that you get. And remember, I'm an engineer by background, so this was a bit intimidating for me to start with. All sorts of arrows and proteins and complicated things going on. But engineers are good at complicated processes, good at solving and understanding complicated control problems. And as a child, I was interested in understanding mechanisms, and I like these cartoons by Heath Robinson. So anybody who knows a Heath Robinson cartoon will know that these often show mechanisms. So the person there is pedaling, and then that's turning this big wheel, and that moves a conveyor belt. And basically, this is a mechanism. Something happens, and then you get a whole series of events, and then you get an output. And actually, we can understand some of these biological signaling processes in a rather similar way. And I know biologists will be saying, no, it's much more complicated. But as I say, engineers are good at understanding complex processes. So I'm going to show you a sort of cartoon of what we found out about how cells sense mechanical loading. And this isn't the only way that cells sense mechanical loading, but this certainly seems to be a really important way in articular cartilage. So when you load cartilage, so if you're walking around and you're squashing the cartilage in your joints, the cells get deformed, they get squashed. And this opens tiny channels on the membrane of the cells called hemichannels, and out from those hemichannels comes ATP. That ATP will then bind to specialist receptors on the membrane of the cells, and that allows calcium to go into the cell and also releases calcium from inside the cell. And this calcium signal then signals to the nucleus, and that activates the production of the new cartilage. So it's a mechanism there through which the cells are sensing that mechanical environment. And then we did the same experiments with cells that didn't have primary cilia, that couldn't express primary cilia because of that genetic mutation. And what we found here is the cells would deform, the hemichannels would open, the ATP would come out and bind to the purine receptors, but then we wouldn't get that calcium response. So the whole sort of mechanism stopped and broke down at that point. So we knew, therefore, that primary cilia were necessary for mechanosensing of in cartilage cells. And this was the work of a very talented postdoc who was working with me, Angus Wong, and he was on a Wellcome Trust grant with Tony Poole and myself, and he was able to show for the very first time that primary cilia did something. They weren't the Loch Ness Monster, they were actually functional in articular cartilage and were involved in sensing that mechanical loading. And this opened up the possibility that we might be able to manipulate primary cilia to control cartilage growth and maybe use that as a treatment for osteoarthritis. What Angus then went on to do was really amazing, and he showed that primary cilia were also involved in inflammatory signaling. So in an osteoarthritic joint, there is a component that is inflammatory, so you get a degree of inflammation in the joint. And Angus was able to show that primary cilia were necessary and were involved in this inflammatory process. So now we've got the potential of being able to manipulate primary cilia to control inflammation. And this was completely new. Nobody had ever dreamed that primary cilia were involved in inflammation. So this was really groundbreaking. And Angus was doing this on a grant from Arthritis UK. So what we've begun to realise is that in osteoarthritis, the environment, the mechanical environment, the inflammatory environment, changes primary cilia function. And that's important for the progression of the disease, driving that degenerative disease forward. And there's now very good evidence that this is what's happening. We can see that some of the functions that are controlled by primary cilia are disrupted in human osteoarthritis. And therefore, potentially, we can get in and treat them with drugs. And the person that really has helped to do a lot of this and is now on the grant working on this is Claire Thompson. So Claire started with me and Paul Chappell as a PhD student and then helped to write a couple of grants, one from the AO Foundation, which she was on, and then has also helped write this grant for looking at osteoarthritis and the role of primary cilia. So what do we know? We've found out that mechanical loading increases the synthesis of cartilage matrix, and it does this through the primary cilium, tipping that balance towards synthesis. But what Claire also showed is that mechanical loading causes disassembly of the primary cilia. So there's a little graph there showing that the length of primary cilia and how they get shorter as you increase the mechanical loading. And that's important because that causes a disruption to some of the mechanisms that are taking place on the cilium. And these are mechanisms that would normally cause cartilage breakdown. So therefore, mechanical loading is good for two reasons. Firstly, it's increasing the synthesis of synthesis of cartilage, and secondly, it's reducing the breakdown. And right at the heart of this is the primary cilium. And actually, we've now begun to realize that this same mechanism also occurs in other tissues, in other diseases. So we've been working with Professor Hazel Screen, whose baby is making a noise at the back there. And Hazel and I have been looking at tendon injury and how primary cilia are involved in tendon injury. And very recently, it's been shown that primary cilia are also involved in atherosclerosis. And the position that atherosclerosis takes in a blood vessel is where the mechanical environment is disrupted. And then you get primary cilia formation and you get the formation of these atherosclerotic plaques. So we think primary cilia are actually involved in a number of different disease conditions associated with changes in the mechanical environment. I'm now going to switch tack a bit, and this is to mention a little bit of work that I'm involved with looking at mechanobiology in cancer. And I'm involved in a few projects on cancer and mechanobiology, but I want to talk about this one in particular. This is a big grant from the European Research Council, and it's called the CANBIL project, and it's led by Professor Fran Boltwell at Bart's Cancer Institute here at Queen Mary University of London. And Fran came up with this crazy idea of being able to bioengineer a human tumour. It was a sort of science fiction idea, but it had a very important and real purpose. The idea was that if we can grow a human tumour in the laboratory, then we can test new drugs and we can understand how cancers grow and spread. And my role in this project has been to look at the importance of mechanobiology in the formation and the growth of the cancer. And I've been working with a very skilled postdoc, Robin Delane-Smith, who's a bioengineer on this project. And the particular cancer that we focus on is ovarian cancer and the metastases that take place in the omentum. So the omentum is a soft, fatty tissue in the abdomen, and what happens is that as the cancer grows, it changes that environment. The tumour begins to stiffen up, and this is associated with the deposition of collagen. And you can see the pictures on the top, the healthy or normal tissue, the black holes are where the fat cells would be, and then in the diseased tumour you get increased deposition of collagen, and that causes a stiffening of the tumour. And we think this change in the mechanics is really important for how the tumour grows and spreads. And then very recently, what Robin has been able to do, and some of the others on the CanBuild project, is to take some cancer cells and the collagen-producing cells, and he's shown that the cancer cells are actually changing the biochemical environment and activating these collagen-producing cells, so that the collagen-producing cells then change the mechanical environment. And it's a particular protein that the cancer cells secrete called TGFbeta. And for me, you can guess what's coming next. The really exciting thing is that primary cilia are involved in this process. So the TGF-beta that the cancer cells secrete changes the structure of the primary cilia and changes some of the signaling that's taking place on those primary cilia, and that's essential for then activation of those cells and the remodeling of the mechanical environment. And here then are some lovely pictures that Robin has taken showing the primary cilia in these cancer-associated fibroblasts, the collagen-producing cells, and how when they become activated, the cilia actually get very short or disappear altogether. So lastly, I'm going to talk about another area of work, looking at the role of primary cilia in stem cell differentiation. And this is important for a variety of bioengineering applications. If we can control how stem cells differentiate into different tissues, and I'm sure you're all aware that stem cells can become fat or cartilage or bone or muscle or nerves. If we can control this process, then we can use it in tissue engineering, but we can also use it for when you put an implant into the body, stem cells will come into contact with that implant, and we need to make sure that those stem cells lay down the appropriate tissue around that implant. And this is work done by a number of people, a couple of postdocs, Rebecca McMurray and Stephen Thorpe, but more recently a PhD student, Melise Dolbe, funded by the Institute of Bioengineering. And Melise showed that as stem cells differentiate into these different types of cells, their primary cilia changed, and that change in the primary cilia was essential for controlling the differentiation of the stem cells. And then what Melise has also done is to go back to our colleagues in biomaterials and look to see how primary cilia are involved in the sensing of stem cells, how stem cells sense different biomaterial surfaces. So we've looked at a bone graft substitute material, and this is a calcium phosphate material, and we have two different materials. One has a very rough surface, and one has a smooth surface. And if you put these into the body, you'll only get bone formation around the rough surface, whereas the smooth surface, you don't get the same bone formation. And what Melise has been able to show is that this is due to the stem cells that come into contact with these biomaterials. They respond to the biomaterial surface, they respond to that roughness and change the structure of the primary cilium and change the differentiation of that stem cell to make it become a bone cell. So this is very exciting because it gives us the opportunity to manipulate biomaterial surfaces to control stem cell differentiation by controlling the primary cilium. So what then is our big overall idea? What I've been trying to say, and I think this is the direction our research is going, is that in disease, that causes a change in the environment, the physical environment, the chemical environment, and this causes a change in the structure and function of the primary cilium. And we think this is important in a range of different diseases. I've talked about it in osteoarthritis, in inflammation, in cancer. We're looking at it in other diseases as well. And this gives us the potential to be able to manipulate the progression of these diseases with drugs or sophisticated biomaterials. And I've called these environmental ciliopathies. So this is a new class of ciliopathy. It's not the genetic ciliopathy that I talked to you and that led to the big interest in primary cilia. But I think this is a new class of ciliopathy which may also have a big change in how we understand cilia structure and function and importance in human health. So what next? I put this picture up here. This is a picture that I took of a primary cilium, and it's my favourite picture because it's a cilium that happens to be in the shape of a question mark. So we've still got lots and lots of questions to answer about the function of this fascinating organelle. We've come a long way from it being thought of as the Loch Ness Monster, And I hope even my colleagues in neuroscience that were originally perhaps a bit sceptical might now believe that primary cilia are there and have a function. But ultimately, what we've got to do is to move from mechanobiology through to what I've called mechanomedicine. We've got to use that knowledge of mechanobiology to develop new strategies for treating diseases and injuries, bioengineering strategies or maybe pharmaceutical strategies. And lastly, one of the things that in universities we have to be very aware of is impact, and I think we can have impact through both education and public engagement, as well as the development of those mechanomedicine strategies. And this is something that I've been very involved with at my time at Queen Mary. So Queen Mary has a fantastic reputation, a fantastic track record for biomedical engineering education. As you've heard, I was part of the Interdisciplinary Research Centre in Biomedical Materials, and we'll be celebrating our 25th anniversary later this year. We now have the Institute of Bioengineering, which is a cross-faculty institute that brings together bioengineers in medicine and dentistry and also across the science and engineering faculty. But perhaps the area of education that I'm most proud of, and I have worked a lot on, is in the undergraduate degree programme. We have at Queen Mary one of the best, if not the best, and one of the biggest undergraduate programmes in medical engineering. And I put this up. This is some of my fantastic project students. These are final year students, and they were about to go into the operating theatre to see how a particular implant was put in that they were working on as part of their final year project. And then there's the area of public engagement. I was a director of public engagement in the School of Engineering and Materials Science, and I have done a lot of public engagement activities in my time. A lot of that has been with the Centre of the Cell, which was headed by Professor Fran Bulkwell, and I have developed various bioengineering interactives that are present in the Centre of the Cell. So that's received 65,000 visitors since it was first set up. And I've also developed a Bionic Man interactive, and that was in collaboration with my colleague there, Elena Damascio. She's the one without the green lights on. And this has gone out to young people at various science festivals, and we estimate that we've had at least 10,000 interactions with young people telling them about the importance of bioengineering and biomaterials. The most important part, perhaps, of an inaugural lecture is the thank yous. I'm not going to go on and on, but I must acknowledge the funding that I've received. And perhaps unusual that I've had funding from the three main research councils. So this is major project funding from the Engineering and Physical Sciences Research Council, the Biotechnology and Biological Sciences Research Council, and the Medical Research Council. And I think this reflects the interdisciplinary nature of my work, and also funding from various other charities and organisations. But my biggest thank you perhaps goes to my research group. Without them, none of this would be possible. So I've mentioned some of them as we've been going along. The ones in bold are still with me. So Claire Thompson, Robin Delane-Smith, and Steve Thorpe, who I've worked with, the postdocs, but also my PhD students, Melise, Sheetal, Dan, Sue, and Gwen. And also the people that were in my group and have now moved on to other things, particularly Angus Wong, who now has a fellowship at Oxford. And lastly, I want to finish on this. As was mentioned at the beginning, I have been part-time ever since I got my postdoc. So when I got my EPSRC Advanced Fellowship back in 2000, shortly after that our first child was born, Jodie, and I went part-time to share the childcare. So I can build a very good igloo and I can cook some very tasty sausages on a campfire. So thank you, Jodie and Rosie and Thomas, and especially thank you, Sarah, for making it all possible. And thank you all.